Hi guys, David here with another science update. So today I'm looking at an article from Nature Communications which is about the connection between traumatic brain injury and improved healing of bone injuries. So apparently that's a well-known connection. So when you have patients who have both a traumatic brain injury and a bone break, apparently the bone break heals better for those patients than if they had not had a traumatic brain injury. And what this paper is trying to do is basically try to really quantify that, make sure that that's true, confirm it, and also try to uncover the mechanisms for it. So as I've discovered from reading biomedical papers, any really good or important work basically involves being really mean to a bunch of rats. So in this case, they picked 30 rats to be mean to. So they basically took the rats and they subsequently randomized into the proximal tibia defect, PTD, and PTD combined TBI, PTD, TBI groups. In other words, they took half of them and they broke their tibias, and the other half they broke their tibias and they gave them a traumatic brain injury. So it's for a good cause, but I just always feel so freaking bad for the animals in these studies. So we'll look briefly at a couple of pieces of data pertaining to actually demonstrating that this improved healing was present in the rats. So one piece of data that they do is they look at the volume of callus formation in the groups that had a fracture. That's the blue dots, and the groups that had the fracture plus the traumatic brain injury, or the TBI. And you can see at 2 weeks, 4 weeks, and 6 weeks, by the time you get to 6 weeks, the callus formation, there's a lot more callus than there is for the just the mice that had the fracture but not the brain injury. And when we're talking about callus formation, what we're talking about is basically when you have a fracture healing, there's a number of steps you go into. The first one is hematoma formation, that's day 1 to 5. And then you have a fibrocartilaginous callus formation from days 5 to 11. And then bony callus formation takes place from days 11 to 28 typically. So this callus is not like the callus on like your finger if you play guitar or something. We're talking about a specific tissue formation pertaining to bone regeneration and improvement. What's interesting is in caption B of figure 1, they're showing the fluid percussion device for experimentally induced TBI. They actually have a device here that is focused on giving a traumatic brain injury to a mouse, but doing it in a highly reproduced way. So we're going to watch just a short clip on how it is that they actually introduce the traumatic brain injury in the rats in a controllable way. And it's a little bit graphic, but I also think it's a bit interesting, so let's just go ahead and watch it.
The overall goal of this procedure is to induce a mild to moderate brain injury in mice to model human traumatic brain injury. This is accomplished by first performing a craniectomy. The second step of the procedure is to attach a hub to the skull over the craniectomy site. Next, a pulse of water is delivered through the hub to induce brain injury. The final step of the procedure is to remove the hub and close the wound.
So after they carry all this out, they basically sew the rats back up, and then they can do all kinds of things like behavioral studies, or they can do the things like are going on in this study that we're talking about today, where they're looking at different factors in the blood plasma and looking at how they heal from other injuries. And basically this has given researchers a really controllable way to study the impact of an induced brain injury. So going into it more, they didn't just stop at quantifying the volume of the callus, they also looked at the bone volume to tissue volume in the area, and they found that again at 3 days, 7 days, and 14 days, again the red dots, the group with the traumatic brain injury, performed much better. They also did some histological studies, so that's looking at actually tissue samples and staining them. And I'm not really any good at histology, but the PTD groups is 7 days and 14 days at the top, and then the group that also had the traumatic brain injury is shown down below. And I think what we're looking at here is basically there's a higher fraction of the white formation is probably the bone tissue formation, and there's more of that going on, I guess, in the TBI group. Though again, that's not really something I'm good at differentiating visually. So this and some other evidence were used basically to show that yes, in fact, this phenomenon that we've seen in humans and in other studies, that the bone injuries heal better when you've also had a traumatic brain injury, are holding true for this group. And so what they did then is a number of mechanistic studies, and it's a little bit more in depth than I want to get into, but what I'm going to do is just briefly summarize the key results that they had, and then we'll look at the implications. So one of the first mechanistic clues they had as to differentiating why it is that the traumatic brain injury induced patients had better bone healing is that looking at the blood of the different rats, they found that the rats that had a traumatic brain injury had elevated levels of small extracellular vesicles. And so these little kind of compartments of material, that there were more of them inside the bloodstream. The second thing they found is that looking inside of these small extracellular vesicles, they found that there were potent miRNAs, which is to say microRNAs, that were associated with osteogenesis. And osteogenesis is the regrowth of bone tissue. So it looked like in the rats that had a traumatic brain injury, they were actually releasing these little vesicles, and in these vesicles were actually signal molecules or RNA molecules, I should say, that contribute to bone regeneration, osteogenesis. And they found that it was the injured neurons in the brain that actually produce these small extracellular vesicles that target bone and osteoprogenitors. So in other words, it was the neurons themselves that sustained damage during the traumatic brain injury that are releasing these small extracellular vesicles that contain these molecules, these biomolecules that encourage bone and osteogenesis. They also found that these small extracellular vesicles had a protein on their surface called protein fibronectin 1, and that actually helps the small extracellular vesicles arrive at the damaged bone site, because protein fibronectin 1 apparently is more effectively accumulates in bone. So to kind of boil some of that information down, we see figure 8. So we see the rat brain injury, or the human brain injury, causes the neurons to get injured. The neurons then release these small extracellular vesicles, and then these small extracellular vesicles have these different microRNA sequences that are osteogenic in nature. And then there's this fibronectin, which is at the surface, which helps the small vesicle actually arrive at the bone tissue. There, the small RNA molecules basically trigger FOXO4 and CBL, which then cause osteogenesis, or the regrowth. So this is basically the mechanism. So this looks like, we've known this connection between the traumatic brain injury and improved osteogenesis or improved bone regrowth, and this looks like actually how it works. So what is exciting about this is that it really probably justifies being mean to all those rats. If you think about it, if we know how this works, then if we wanted to improve bone healing, then we don't necessarily have to give someone a traumatic brain injury, which they would never do. That would be crazy. Instead, what we could maybe do is look for ways to maybe make some of these extracellular vesicles with the right contents artificially. And maybe in the future, if you get a bone break or something like that, especially a really major one, perhaps they could just directly inject something like this structure and accelerate your bone healing. Well, this has been another Science Update. Thanks so much for watching. If you enjoyed the video, please go ahead and like and subscribe, and I hope you'll join me again in the future soon. Bye.